Hello and welcome to our last video of our discussion series about solutions. In this video, we will be talking about colligative properties. The presence of solute gives a solution different physical properties than the pure solvent. But in the case of four important properties, it is the number of solute particles, not their chemical identity, that makes the difference. These colligative properties are vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure. Most of the effects are small, but they have many applications, including some that are vital to organisms. So we predict the magnitude of a colligative property from the solute formula, which shows the number of particles in solution and is closely related to our classification of solutes by their ability to conduct electricity. So the first classification is electrolytes. When we say electrolytes, this separates into ions when it dissolves in water. So a strong electrolyte dissociates completely, while a weak electrolyte dissociates very little. An aqueous solution of an electrolyte conducts a current because the solute separates into ions as it dissolves. And the non-electrolytes do not dissociate to form ions, so each mole of dissolved compound yields 1 mole of particles in solutions. So compounds such as sugar and alcohol do not dissociate into ions at all. They are non-electrolytes because their solutions do not conduct a current. So here is an illustration of the non-electrolyte, a weak electrolyte, and a strong electrolyte. And here are the colligative properties that I have mentioned in the previous slide. We have the vapor pressure lowering, boiling point elevation, freezing point depression, and osmotic pressure that we will be discussing in a while. For vapor pressure lowering, a volatile solute readily escapes into the vapor phase. A non-volatile solute, however, does not readily escape, so it has a negligible vapor pressure at a given temperature. So we have to remember that the vapor pressure above a solution of a non-volatile solute is lower than the vapor pressure of the pure solvent. So why is the vapor pressure of a solution lower? The fundamental reason for this lowering involves entropy, specifically the relative changes in entropies of vaporization of solvent versus solution. So a liquid vaporizes because a gas has higher entropy, and in a closed container, vaporization continues until the number of particles leaving and entering the liquid phase per unit time are equal, as shown here in the illustration. That is, when the system reaches equilibrium. But as we said, the entropy of a solution is already higher than that of a pure solvent. So fewer solvent particles need to vaporize to reach the same entropy. With fewer particles in the gas phase, the vapor above a solution has lower pressure. The next colligative property would be the boiling point elevation. So in the boiling point elevation, a solution always boils at a higher temperature than the pure solvent. So this colligative property results from the vapor pressure lowering. So the boiling point elevation is proportional to the molality of the solution. So we have this formula. So the change in the boiling point or the temperature is equal to Kb, or the molal boiling point elevation constant, multiplied by the solution molality. So remember that molality is used because it relates to mole fraction and thus to particles of solute, and it is not affected by temperature. So in your book, you can find in table 13.6 of page 544 the different values for molal boiling point elevation constant. And then for freezing point depression, a solution always freezes at a lower temperature than the pure solvent. So the freezing point depression is proportional to the molality of the solution. So we have the formula: change in freezing point or change in temperature is equal to Kf, which is the molal freezing point depression constant, multiplied by the molality of the solution. Again, in your book, you can find the values for the molal freezing point depression constant for different substances or different solvents. So here is the table I was talking about earlier. So example, in determining the boiling and freezing point of a solution, we add 1 kilogram of ethylene glycol anti-freeze to 4,450 grams of water in your car's radiator. So what are the boiling and freezing points of the solution? So we have to in order for us to solve this problem, we need to find the molality of the solution and then calculate the boiling point elevation and freezing point depression. So remember that we have to convert first the mass of the substance into moles, and by converting, we will arrive at the amount of substance 16.1 moles of ethylene glycol. And then after that, we have to find or we have to compute for the molality of or the concentration in molality of the solution, wherein we divide the amount of substance by the mass of the solvent or the mass of the solution, which is 4.450 kilograms. So in here, we converted grams to kilograms, and then we will arrive at the concentration 3.62 molal ethylene glycol. And then to find the boiling point of the solution, we just substitute the given value the computed value 3.62 molal and multiply it by the boiling point or the molal boiling point elevation constant of water, which is 0.512 degrees Celsius per molal, and that would be equal to 1.85 degrees Celsius. After that, to compute for the boiling point of the solution, we have to add the computed value 1.85 degrees Celsius to the boiling point of water, which is 100 degrees Celsius, and we will have the new boiling point of the solution, which is 101.85 degrees Celsius. And as for the freezing point depression, again, we will multiply the concentration of the solution, which is 3.62 molal, by the freezing point or by the molal freezing point depression constant of water, which is 1.86 degrees Celsius per molal, and that would be equal to 6.73 degrees Celsius. And since this is freezing point depression, we will subtract the computed value from the freezing point of the solvent water, which is 0 degrees Celsius. So 0 degrees Celsius minus 6.73 degrees Celsius, that would be equal to negative 6.73 degrees Celsius, and that would be the new freezing point of the solution. Another colligative property would be osmotic pressure. As for the osmotic pressure, recall from your general biology class that osmosis is the movement of solvent particles from a region of higher concentration to a region of lower concentration through a semi-permeable membrane. So solvent will always flow from a more dilute solution to a more concentrated one. That's why osmotic pressure is the pressure that must be applied to prevent the net flow of the solvent. So osmotic pressure can be calculated by the formula MRT, where M is the molarity, R is a constant, a universal gas constant, and then T is the Kelvin temperature. Example, biochemists have discovered more than 400 mutant varieties of hemoglobin, the blood protein that carries oxygen. A physician dissolves 21.5 milligrams of one variety in water to make 1.50 milliliters of solutions at 5.0 degrees Celsius. She measures an osmotic pressure of 3.61 torr. What is the molar mass of the protein? Based from the problem, we know that the osmotic pressure is equal to 3.61 torr, and then we also have the universal gas constant and the temperature, which we need to convert to Kelvin. And then we convert the osmotic pressure from torr to atmosphere and the temperature from Celsius to Kelvin, and then use the equation we presented earlier to solve for molarity. Then we calculate the amount of hemoglobin from the known volume, which is 1.50 ml, and use the known mass 21.5 milligrams to find the molarity or the molar mass of the protein. So for our solution, it would be like this one. Again, we convert the pressure to atmosphere, and then we manipulate the formula. Molarity is equal to osmotic pressure over universal gas constant times the temperature in Kelvin. So if we substitute the given values, the converted values, we will arrive at the answer 2.08 times 10 raised to negative 4 molar. And using the molarity, we can now determine the amount of the substance by multiplying the molarity of the substance or the concentration of the solution by 0.00150 liters, and we will arrive at 3.12 times 10 raised to negative 7 moles. The molar mass of hemoglobin after changing milligrams to grams, so we have 0.0215 grams over the computed amount of substance moles, and then we will arrive at the answer 6.89 times 10 raised to 4 grams per mole. So here's an illustration of the movement of solvent from a dilute solution to a more concentrated solution through a semi-permeable membrane. So remember that osmotic pressure is the pressure that prevents the flow of additional solvent into a solution on one side of a semi-permeable membrane. Remember that in your general biology class that you have also discussed the types of solutions based on the osmotic pressure. One of those types of solutions would be isotonic, wherein two solutions with the same osmotic pressure are said to be isotonic. And then for a hypotonic solution, it has a lower osmotic pressure than the body fluids, and the concentration of particles outside the cell is lower than the concentration of particles inside the cell. Thus, water diffuses into the cell, so the cell swells and eventually bursts. For red blood cells in our body, this swelling and rupture is called hemolysis. And then the last type of solution according to osmotic pressure would be the hypertonic solution, wherein it has a higher osmotic pressure than body fluids. In that case, the concentration of particles outside the cell is higher than the concentration of particles inside the cell. Thus, the water diffuses out of the cell, which causes the cell to shrink. And then this process is called crenation. Again, these are the electron micrographs of the cells in different conditions. Here is a cell in a dilute salt solution. This is an isotonic solution. And then cells in a distilled water, a hypotonic solution. And then cells in a concentrated salt solution, which provides a hypertonic environment. And as for the application in health and medicine, we have here dialysis. So as we all know, dialysis is also a process that involves the selective passage of substances across a semi-permeable membrane called a dialyzing machine. In dialysis, however, water, small molecules, and ions can travel across the membrane, and only large biological molecules like proteins and starch cannot. So in the human body, blood is filtered through the kidneys with the process of dialysis. So here is the process of dialysis. Each kidney contains over 1 million nephrons, tube-like structures with filtration membranes. These membranes filter small molecules like glucose, amino acids, urea, ions, and water from the blood. So useful materials are then reabsorbed, but urea and other waste products are eliminated in the urine. So when a person's kidneys are incapable of removing waste products from the blood, hemodialysis is used. A patient's blood flows through a long tube connected to a cellophane membrane suspended in an isotonic solution that contains sodium chloride, potassium chloride, and sodium bicarbonate, and glucose. So small molecules like urea cross the membrane into the solution, thus removing them from the blood. So red blood cells and large molecules are not removed from the blood because they are too big to cross the dialyzing machine. So remember that when a patient's kidneys no longer function properly, periodic dialysis treatment are used to remove waste products from the blood. Again, blood is passed through a dialyzer which contains a membrane that allows small molecules to pass through, thus acting as an artificial kidney. So each treatment takes several hours. Patients usually require 2 or 3 treatments per week. And that ends our discussion series for solutions. Thank you for listening.